All right. So good morning, everyone. Welcome to our weekly orthopedic surgery grand rounds. Today, I have the pleasure of introducing one of our residents, Dr. Stephen Garcia. So Dr. Garcia is originally from Mira Loma in California. He did his undergrad at UC Riverside, where he did biomedical engineering, and then also received a master's after completing his undergraduate work. Following his time there, he came to UCSF for medical school. And then we were fortunate to have him match with us in our orthopedic surgery residency program. He did the 6-year originally research track. His clinical work has been great. He is applying into pediatric orthopedic surgery this year and is looking forward to a bright future there. His research work has really been just beyond exceptional, so much so that he extended the 6-year program into a 7-year program, taking a second lab year just because his work was going so well. So Dr. Garcia has worked with the Feely Liu lab. He has done remarkable work on muscle stem cell function and understanding different properties of stem cells as they relate to muscle recovery. His work has been funded by the OREF, by the NIH. He has received basically every award that I think he's ever tried to get. He's received awards from the OREF, from ASES, AOSSM, a number of really prestigious awards for any investigator, much less as a trainee. And so, Dr. Garcia, thank you for coming to Grand Rounds today, and look forward to your talk on musculoskeletal stem cell heterogeneity and tissue function.
All right. Thanks, Dr. Lanzone, for those kind words. All right, this is going to be a dangerous talk since there was no coffee and we're talking about science, but we'll try to keep everyone awake here. Okay, so I'm going to talk to you a little bit about some of the work I've done with musculoskeletal stem cells and their diversity and heterogeneity and how that relates to a little bit to tissue function. So I don't have any real disclosures, so I decided to humble brag a little bit with some of my grants, but they do relate to stem cells and diversity, so we'll kind of go through this as I go through the talk. And my objectives for today are to really convey the importance of heterogeneity and diversity in biology, and then to demonstrate that stem cell heterogeneity drives tissue function, and by going through two short stories, one in satellite cells and one in fibroadipogenic cells, and then intervene a little brief history of single-cell RNA sequencing, because you're going to be seeing a lot more of this when you read papers in orthopedics, and then hopefully by the end inspire some collaborations in MSK research. So I just wanted to start the talk by thinking about diversity in biology, and when I was thinking about that, I was thinking about the animal kingdom, because my mind is drawn to. And if you think about plankton, they're one of the most diverse collection of organisms on this planet. They're in every biome. The air you're breathing right now is about 60% produced by marine plankton. And they're so diverse, in fact, that in 1961, Dr. Hutchinson wrote this famous paper that's still quoted today that said that they're too diverse even for their environment. Like the nutrients just couldn't support them. Well, we know that over the next 60 years of research that's actually not true. It's because the diversity of their natural environment is so rich that they can be so diverse as well. And I think this is true in my life as well. I would be remiss to not thank Michael Davies, who's been such an important figure in my life, a tremendous colleague and friend, and I definitely wouldn't be here standing in front of you, so I just want to acknowledge him and embarrass him even though he's not here. And then I think as I go through this talk, all of these people who have been mentors and mentees and colleagues have shaped who I am as a scientist, and I want to call them out as I go through the talk. But hopefully it'll be obvious by the number of people who I've worked with that this has all been important. So thank you to everyone in the Pomerantz Feely Lab and all of our collaborators here at UCSF and also at SFGH. And thank you to everyone who's provided samples for this study from their patients. So it does mean a lot. So my story is not unique. I think diversity in scientific work is very important. It's also been shown in a lot of recent literature to produce better science. So specifically, ethnic diversity and gender diversity produces more novel and higher impact science ideas, and objectively, they get into better papers and with more citations. So in our lab, just to sort of improve on this model, Dr. Feely and I have been awarded a diversity grant to continue this on, and this has really been spearheaded by Augustine Diaz, who's actually in the audience today. We're lucky to have him, who was in the lab and kind of brought in underrepresented students and undergraduate to get them involved in musculoskeletal research from an early onset to hopefully retain more diversity in our field. So my first just quick take-home point is diversity produces better science and better scientists. And just to think about this when you're forming your groups and your teams, because this really does matter. And now to get into a little bit of the science, to draw a little parallel to diversity of people, I've been studying stem cell heterogeneity since med school. And my first foray into that was within the Pomerantz lab. And these 4 people have been pretty integral to that in the lab of Jason Pomerantz, also Stan Tamaki, Alvin Wong, and Emily Beret, who have all been instrumental in that. And what we were interested in studying in that lab is how skeletal muscle regenerates after injury. So if you allow me to show you this little cartoon, if you look at a single myofiber, satellite cells exist on the edge of the myofiber. And what these cells do is when muscle is injured, they become activated, they migrate to the injury area, proliferate, and then can differentiate to form myocytes, but then fuse to form a new myofiber. And these satellite cells were only recently discovered in 1961. And Dr. Mara at that time thought these cells had a big nucleus compared to their cytoplasm, so maybe they were a dormant cell that could be responsible for the paradox of muscle regeneration in adult tissue. We since know that since then, just like in the frog, the mouse can regenerate, and it is because of these cells. So I was lucky to join the Pomerantz lab when they had just been one of the first few labs to show that human muscle also has these satellite cells, and they're responsible for muscle regeneration. So if you take a muscle fiber and you bring out a single myofiber and put it in a dish, it kind of looks like this. And you can stain for laminin, which is the basement membrane that surrounds the tube. And then if you stain for PAX7, which is the conical marker of a stem cell in muscle, or a PAX7 positive muscle satellite cell, you can see that these little kind of teal colored cells, because it's green plus blue, exist throughout the length of the fiber. And if you isolate these cells from human tissue and put them in a immunodeficient mouse, and then injure the mouse, you can grow human muscle in a mouse. So you can have a kind of humanized mouse muscle. And after about 5 weeks, you see a lot of these spectrin positive cells and also laminin AC positive, spectrin positive fibers and laminin AC positive cells, indicating that you can regenerate human muscle in mice just with these cells. So when I got involved with the help of Stan, we further showed that these cells are true bona fide stem cells, meaning they do 2 things. 1, they self-renew, so you can make more of the same cell. And 2, they will differentiate and then form the tissue type that they're in. So in this case, regenerate skeletal muscle. So what we did was we took human tissue, isolated the stem cells, put them in a mouse, and then you wait about a month, and then you could take the cells back out and put them in another mouse. And if you look at flow cytometry data from this, you can see that we can pull out CXCR4, CD56, CD29 positive stem cells. And then if you put them in mice in a primary or secondary transplant, you can grow human muscle as indicated by human dystrophin positive fibers. And this is in like a cross-section immunofluorescence image of a mouse muscle. So you're growing human muscle in a mouse. And one thing that we were curious about during this work is that there was something specific about some of the cells that led to better engraftment. And we know from past studies, like from Brack's lab and from others, that their stem cells can undergo different differentiation or self-renewal pathways. And we wanted to know, is the PAX7 positive cells, are they all homogeneous? Or in fact, is this a heterogeneous population that some of the cells are more stem-like and do these stem cell things? So we were lucky at this time, which is in about 2016, that technology had advanced to where we could analyze cells at the single cell level to really resolve this heterogeneity. And to kind of go over that, I'm just going to give a brief history of how we have typically evaluated RNA, for those who may be unaware. But in the 80s, PCR was developed, and then that led way to qPCR, which, depending on how much you wanted a pipette in a certain day, you could do 1 to 20 genes. But you basically are looking at a gene very manually, and you get a bar graph where you compare a control to an experiment, and you tell like how different the genes are between those two. And over the next decade, this led to bulk RNA sequencing, which is a similar thing, but now you're looking at the whole genome. So in humans, that's 18,000 genes. And you get groups of gene changes. So you can really look at like pathways of genes that are changing or ontologies of things that are changing. But still, with both of these, you're limited to your one sample. So you either have a mixture of cells or you have a collection of cells from a tube, but you can't resolve single cell differences. So then in the late 2000s, single cell RNA-seq was a new technology that was being developed. And really what you got is the same amount of genes, so 18,000 genes, sorry, it's cut off, but for each individual cell. And this way you could look at how the cells, even in a solution, were different. So in the early 2000s, the late 2000s, this was a very labor-intensive process, with the first studies being published in 2009. And this was, you had to do 1 to 100 cells at a time. And this was very expensive because you imagine you're sequencing every cell individually. But then I just wanted to point out that I was very lucky that I joined a lab that was interested in this right when this was becoming commercially available, with 2 techniques called DropSeq and 10x genomics. And what they did was really allowed for parallelization with a microfluidics device that allowed for barcoding of the cells. So you could do thousands of cells at the same time. So what we were interested in is looking at this in human muscle. So again, with the help of Stan and Emily too, we looked at human muscle, so we could digest the muscle that we get from patients. We could flow cytometry sort to enrich for our stem cell population. And then we could perform this microfluidics device technology where you do RNA amplification and sequencing all within this little bubble that contains one cell. And then you get data that looks kind of like this 2D plot, and you could do gene analysis. So what that looks like is this 2-dimensional UMAP plot, which looks very colorful, and each dot represents a single cell. And I think the first thing you generally do, and when you get at this in lab, is you show it to your lab mates, and it kind of acts as a Rorschach test, so they can tell you what your favorite animal is. But I like to think it looks more like a piece of art, where pointillism is a French artist technique, and it's actually named, one of the main toolkits is named after that painter, Georges Seurat, who this group invoked the analogy that it looks like this dots form a spatial pattern of a pointillist painting. So if you're interested, you should look up more about him. But also, if you're a super nerd like myself and my fiance, you can go to Père Lachaise Cemetery and actually see where his family crypt, to get more involved in bioinformatics. It's sort of a pilgrimage. But really what this data is telling you is that every dot on this spot, on this space, is a single cell. So here you're looking at 60,000 plus cells all put together. And each cell has 18,000 genes. So if you can imagine the biggest Excel document you'd ever work with, this is bigger. And each cell, the cells are clustered according to their whole transcriptomic similarity, and the colors represent cells that are most alike. So like these blue cells are most alike each other in the population. And I think it's pretty important to know how to read this type of graph, because even in some orthopedic journals, we're starting to see a lot of these graphs. So I'm going to go through that by showing you a quick example of how we get to that plot, and that's through principal component analysis, which Halverson can probably explain better than me, but I'll try to explain. And that's really a mathematical tool for determining and displaying variance or variation in your data. So we're going to use a random sample data set of UCSF ortho to try to cluster us according to our similarity. And this by no means assumes anything about anybody. I just picked random things. So some things you could put along a variance would be something obvious that you could probably guess what this is, which would put it in a bimodal distribution. And this could be gender. So we would separate it into 2 groups, one source of variation for this data set. Other data sets could be, or other principal components of variation could be related to each other. So where you get this linear graph, and that could be related to years in ortho or number of surgeries performed. And then you could use things that are maybe not related or related to allow you to see outliers in the data, where Feely and I maybe have the most basic science interest or FAP interest. Unless you included Michael, he might be over there in the chart. But really what this all allows you to do is display your variance on different plots. And then what you are attempting to do is collapse all of that data into one 2-dimensional plot. So really, if you thought of stacking all these PCs and tracking where each sample sat in these 2-dimensional plots, then you do a nonlinear reduction to get to a UMAP plot. So that's a 2D plot, which is a representation of the data. And then maybe in ortho, what all this underlying variation collapsed down to is what we're interested in and what are the types of patients that we treat. So maybe we cluster together by our specialty or the area of the body that we work on. So again, understanding how to read this is important, but also understanding its limitations is important as well. And again, each dot's a cell, and there's 18,000 genes for each dot. But when you collapse down from all those PCs, you get a distortion. So it's important to not make inferences between populations that are close together. If you were looking at a three-dimensional globe of Greenland from the top down, it might look like this. But if you put it on a 2D space, it would be distorted, and you couldn't really say the topography of Greenland looks like this, where the distances you can measure on a 2D plot. It's more of a 3D-dimensional structure. So for us, back to the science a little bit, we were interested in this population of caveolin-1 positive PAX7 cells. These are all PAX7 positive cells. You also see we got some other cell types in here, like myocytes or smooth muscle cells. And the reason we were interested in this population is that we noticed that the gene expression of these cells was at a quiescence marker, and I'll explain a little bit about what that means. But we noticed that this CAV1 expression pattern was similar in a lot of different muscles that we tried, not just the legs. So if you look at the vastus or the rectus or even the abdominis or pec muscle, there's heterogeneous expression denoted by the higher blue or deeper blue color in these UMAP plots. And also within individual muscle tissue, if you look at PAX7 expression, which again are those stem cells, sorry if it's a little bit hard to read, but you can see a red positive and red negative caveolin-1 positive stem cells. So the reason we were interested in this is because quiescence plays an important role in regeneration of tissues. And all quiescence really is referring to is a cell that can exit the cell cycle and stay sleepy until it needs to be activated. And in muscle, we know that this pathway is very well defined, especially in the mouse, and that the maintenance of the regenerative potential is really dependent on these quiescent PAX7 positive satellite cells that don't express these other clinical markers of muscle differentiation. So with the help and collaboration of Andrew Brack, Emily and I were able to investigate these cells further, because you could just say, well, okay, all you have is mRNA data, but does this matter functionally? So with this RNA data, we were able to and protein data, we were able to isolate CAV1 positive or CAV1 negative muscle stem cells and put them in a dish. And the thing we noticed is that the CAV1 positive cells remained small and round, which is a typical stem cell characteristic in a dish in plastic, while the CAV1 negative cells like to spread out and differentiate, as indicated by the percent of them that started to differentiate within a few days. And then we looked at those downstream clinical markers of muscle differentiation, like we talked about, and we found that the CAV1 negative cells, this is probably really hard to see, sorry, that MyoD expression is up. So they are expressing this master transcription factor of muscle differentiation, and they're going down a muscle pathway, while the CAV1 positive cells have no MyoD, but a lot of PAX7, indicating that they're still a stem cell even after several days in the dish. So with that, you could say, okay, that was just in the dish, but what about in vivo? I would say you're a good scientist. And I would say that we then took those cells and then put them back into a mouse. So just like we did with the original experiments, except we separated the cells by CAV1 expression. So we have CAV1 positive or CAV1 negative cells. And really the CAV1 positive cells conferred the most robust engraftment and growth of the skeletal muscle in a mouse. And also interestingly, they produced the most self-renewal as well. So you have a lot of these PAX7 positive human cells in mice as well. And that's indicated by all these little red and green cells, and also the bar graph. And then you're a crafty scientist and you say, well, are they truly stem cells? And I say, okay, we did that too. We took these cells and then did serial transplantation of just the CAV1 positive versus the CAV1 negative cells. So we took cells from human muscle, put them in a mouse. Again, you wait a month. You isolate them, you put them in another mouse, and then you can analyze it. And really, only the CAV1 positive cells were able to be transplanted multiple times, indicating that they're truly the real stem cell. And that's evidenced by CD56, CD29 positive cells that you could get out of that second mouse with flow cytometry. So my take-home point number 2 is that human satellite cells are heterogeneous. They're not a homogeneous population. And we were able to utilize this brand new technology with single-cell RNA sequencing to show that, and that these cells are also quiescent stem cells that have functional differences and importance for skeletal muscle regeneration. So the next topic, so that was great in the sense that muscle regenerates well, but what happens, we're interested when muscle doesn't regenerate well. So luckily I was able to get in touch with Dr. Feely and Dr. Liu and also Michael to study what happens when, you have a chronic injury and muscle becomes fibrosis and fat infiltration. And they were looking at these interesting cells called mesenchymal progenitors, or FAP cells. And these are in contrast to the satellite cell, which was found in 1961 or talked about in 1961. This cell had just been discovered, so to speak, in 2009. So a relatively new cell. And these cells, if we go back to our cartoon, they're also sitting on the top of the myofiber, and they're doing things during muscle regeneration. So if the muscle gets injured, these cells talk to satellite cells and also immune cells, and they lead to a process that leads to normal, healthy muscle. But when the environment is deranged, these cells can differentiate it to fibroblasts or adipocytes, and that leads to chronic hallmarks of pathologic muscle degeneration like fibrofatty infiltration. We see this in developmental disorders like muscular dystrophy or chronic injuries like rotator cuff tears. So we knew that this FAP population that expresses PGY4 alpha has multiple heterogeneous roles, but we don't know, is it one cell type here, or is this just a bad marker and we have multiple cell types? So we wanted to understand what are these subpopulations doing in healthy tissue and injury, and then how are these lineage decisions made to form different terminal phenotypes. So again, we took human muscle, and this time we designed it so that we could take healthy and injured muscle from the same patients. So patients that had rotator cuff injuries, either partial or full thickness tears, we took the injured muscle during the rotator cuff repair and also a healthy sample of the deltoid, and then also compared that to some healthy hamstrings and did flow cytometry to enrich for the FAPs, and then did single cell RNA-seq analysis. And this is a lot of work with Justin and Augustine and also everyone else in the lab. And what you get out of that is another 2D UMAP plot. And really what we see here is a few different cell types. And the one we're interested in is this PGY4 alpha positive cells. So here we're looking at 35,000 cells, but the majority of them are this population. So we can take that out and re-subset it on the computer. And you get a UMAP plot that looks like this. And the different colors, again, represent these different clusters or different cell types or states. And we have 25,000 cells from 8 patients and 14 samples. And what we see based on their gene signatures is that we think we have hypothesized based on the mRNA that we have stem populations or an activated stem cell population, and then also what looks like a pre-pathogenic looking cell types. So adipogenic cells marked by these two markers, which are CD10 and DLK1, fibrogenic cells marked by CD55, and then maybe a mechanosensitive population marked by piezo, which is a mechanosensing channel. So then again, you could say, okay, this is just mRNA, but do they express the protein, which is what really matters, right? That's what gives you the phenotype. So what we're able to do is do high-dimensional protein quantification, and the way we look to do that in the lab with Hannah as well as Justin and Augustine is by looking at spectral flow cytometry, which is sort of like the next generation of flow cytometry that allows you to look at up to 40 different proteins at once. And the way that works is you're able to combine all of your antibody cocktail into one mixture, and then you get a plot that looks like this where you collect light from 46 different channels, and you get a huge kind of complex waveform. But with the computer, you can do a Fourier transform, which allows you to pull out individual wavelengths of light, and you can look at all the different proteins that you have stained for. And for us, we were interested in those 3 different type of populations. So a stem cell population or progenitor population, a fibrogenic or an adipogenic cell for those lineages. And if you look at kind of a lot more dots, if you're into dots, flow cytometry and is also a good way to look at these. And we found that there are these discrete populations of FAPs that express these markers, indicating that we have distinct subtypes in human muscle, just in healthy muscle. So in order to look at how these cells can differentiate it, if you put human FAPs in a dish and put them in different environments, they'll differentiate into different cell types. So if you put them in growth media, they'll stay sort of like a stem cell. If you put them in adipogenic media, they'll form fat. If you put them in fibrogenic media, they'll form this collagen sheets. One issue with studying human data is that you can't take human samples from a patient over a course of time, which probably wouldn't pass the IRB. So one thing you have to do is look at a single time point and try to make inferences for what those cells could be doing along a lineage. And the one way you can do that is sort of kind of predict the future. And we know there's a technique that was developed that looks at the natural properties of RNA. So when RNA is produced in a cell, it's called nascent RNA or pre-spliced. And that means, if you think back to undergrad, it has both introns and exons. And as that RNA matures, it goes through a bunch of steps, which leads to all the introns being cut out. And that's called spliced RNA. So a current cell that's been matured will have a lot of spliced RNA for what defines it as its identity. But as it produces more pre-spliced RNA, you can think of that's where the cell wants to go. It's the new RNA. So if you analyze the pre-spliced and spliced RNA for all of our cells that we showed in that prior two-dimensional plot, you get something that looks like this. It's kind of like a weather map. And this qualitatively tells us where these cells could be heading. So if we show a cartoon represented, we have this activated stem cell population that looks like it has two pathways towards this adipogenic or fibrogenic population. And then there's some back and forth between the progenitor and the activated population. So this is a way to sort of gain an hypothesis of what's going on. But to test that, we wanted to look at how FAPs transition in the dish. So again, we took human FAPs and put them in these different culture medias, but this time only for 5 days. So they're not going to really produce that terminal cell type, but it'll be a transitioning cell type that may go through these two things that we hypothesize. And if we think that if we put them in a adipogenic media, they'll produce more of the CD10 molecule. And if they go in the fibrogenic media, they'll produce more of this CD55 molecule. And we see that if you just take an immunostain of these cells produce a lot more CD10 in the adipogenic media, while they do not produce any in the fibrogenic media. And conversely, for the CD55 stain, they produce none or very little in the adipogenic media and a lot in the fibrogenic media. And really, if you quantify the ratio of these, this is kind of like the crux of it, that you get more CD10, CD55 negative cells in the adipogenic media, meaning they're going through this intermediate pre-adipocyte pathway. And if you put them in the fibrogenic media, you're getting more of the CD55 positive, CD10 negative cells, indicating that they're going through this pre-fibrogenic pathway. So really what this allowed us to do is to kind of confirm that hypothesis that we made from the computer and show that the stem and progenitor cells go through these two different lineage types through this intermediate cell type. Sorry. Yeah. Okay, but again, this is just in the dish. So what about in vivo tissue? So we wanted to look at the gene drivers of this process, but in muscle that we know is going through this fibrofatty degeneration. And if you think back to how we designed the experiment, we are able to have both partial thickness and full thickness tear tissue. And really, if you look, everyone's looked at enough of these MRIs, but the degenerative tissue that you see is really in the full thickness cohort, where you get a lot of fibrofatty degeneration that you can quantify with Cutal A classification. Whereas in the healthy, or even in a lot of the most of the partial thickness tears, we don't see this. So if we look at the bulk changes of the genes that are different between full thickness tear muscle and healthy muscle from the deltoid, or even from the partial thickness tear group, you can display that on what's called a volcano plot. And this really is a plot that shows the statistical significance for gene differences. So a log to fold change is how much the genes are different between the two samples. So if it's the dot is on the left, then it's more that gene is more increased in the injury, and if the dot's more on the right, that's more increased in the healthy tissue. And in the chronic injury full thickness tear muscle, we see a lot of markers of activation like FOS genes, or markers of having more progenitor cells like THY1. And then also what you see is loss of this DLK1 protein, which we thought was interesting because this protein also marks the pre-adipogenic population. So if we look at DLK1 again in more detail, we see that it expresses a lot of markers that mark a pathway of adipogenesis. And if you stain for this protein in human tissue sections, we can see that even that this protein is very heterogeneous in its expression as well. So we have PGY4 alpha positive cells, which are in red, that express both DLK1 or don't. is indicated by red cells that are green or red cells that are not green. Another thing that was interesting about DLK1 is that these have been studied extensively in the farm animals, so especially in sheeps. The sheep on the right has a much bigger rump, and that's because there's a naturally occurring mutation that is upstream of the DLK1 gene in the promoter region that leads to increased amount of DLK1. So these lambs actually have a lot of muscle but no fat, which is, if you think about the converse of what we see with chronic injuries in rotator cuff tears. And they're called calipige, and this is actually named after this statue, which is called Venus calipige. And you can probably guess why they chose to name this, but it literally means Venus of beautiful butt. So we thought that could be interesting, right? That's like the opposite of what we see in chronic injury. So could we harness DLK1 to prevent this pathway from happening in human muscle? So for the first thing we wanted to do was to find out if the protein is decreased in chronic injury. So again, with Hannah and Augustine, we looked at human muscle digests and performed full spectrum flow cytometry of freshly sorted cells. And this again lets us really track what the individual FAP populations are doing. And if we look just at the DLK1 only expressing cells, in the healthy in the healthy muscle, there's about a 20% expression of this marker on each individual FAP. And in injury, this is about 40% less, or around 11%. In this one particular sample, it was much less in this flow cytometry representation. But so this data confirmed that not only is the protein decreased, is the RNA decreased, but the protein is decreased as well. And if we look at the differences in the subpopulation of DLK1, back at the difference between partial thickness and full thickness tears, we see that there's a decrease in the DLK1 population in the injury, indicated by the orange dots, and an increase in the CD55 population indicated by the orange dots as well in injury. And this goes back to that ratio that we showed in the in vitro dish, also holds out true in vivo. And then what we did was look at the genes that are changing within this specific population, and we see that there's less cells, but there's also much less DLK1 expression and much less expression of SOX9, which prevents adipogenesis in this pathway, and more expression of master regulators of beige and white fat production. So the next thing we thought was, okay, that's great that we found this difference, but what happens if you add it back? So with the help of Miguel, we've been looking at how DLK1 protein can inhibit this process of adipogenesis to hopefully give us a way to prevent or ameliorate fatty degeneration. So if you put human FAPs back in the dish and you treat them with adipogenic media with a control, so PBS, so that's just a phosphate buffered saline, you get a lot of fat, which is green. And if you add recombinant DLK1, you get much less fat. And that's borne out if you quantify that as well, and it's about almost half less. And we think that is because we're getting a reduction in the amount of pre-adipocytes that are forming, by evidenced by the number of nuclei here, as you can see as well in that picture. So our take-home point number 3 is that we have an updated model of FAP lineages where the human FAP population is heterogeneous, and its heterogeneity is linked to stem cell function. And really that allows us to track how these cells are going through 2 different pathways and what can be involved in forming FAP-related fatty infiltration. And that loss of DLK1 leads to this fatty degeneration. If you add it back, at least in the dish for now, that can prevent this process from happening. So that's for that story, but I wanted to say what other things that we can answer sort of with this technology and some of the work that I've been lucky enough to work on at UCSF. And one of the things with Dr. Wong and Dr. O'Connell at here is looking at gender differences in ACL. And we have been harvesting ACL remnants and looking at using the single-cell RNA-seq technology to look at what could be driving the increased incidence of female versus male ACL tears, especially given that they don't seem to hold true after reconstruction. And this is also with the help of Dr. Westreich and Dr. Zimmel providing samples. And then another collaboration that you can use to look at with this technology is how FAPs are involved in muscle or in fracture healing. And we're working with Dr. Miklau, Marcusio, and a group in France, Dr. Colneau, along with the hand group to look at how FAPs are involved in fracture healing. And as far as what I'm interested in continuing with this work, back with the help of Stan, who's been helping kind of guide this project, is looking at spatial protein analysis. So all of the technologies that I presented so far, you lose your spatial encoded information because you're digesting the tissue and then doing an analysis like in flow cytometry or the single cell RNA-seq. But if you want to know really what's happening in vivo, you need to know where those proteins are. So this is a technique that's using mass spectrometry imaging to allow you to look up at least 40 plus proteins at once. And it looks like this. It kind of looks like it's going to harness the power of the Death Star, but it's making this ion beam to create an image that's very colorful. And you can look it up to 20 proteins so far in human muscle. So this is a sample image of that. And our work is kind of continuing along this DLK1 question and story, where we're looking at how to unravel how DLK1 is actually functioning within human muscle and how it functions in development versus adults, and how the cleavage of this protein can lead to two different phenotypes, where we can have prevention of pre-adipocyte differentiation, but also prevention of differentiation of these cells. And finally, to understand how I told you that there's two pathways that we've sort of delineated now that FAPs are going through, but how are they doing this and how are they choosing what cell to become? And we're looking at another technique that looks at the chromatin of the cells with single cell attack-seq. So hopefully over this talk I was able to convey kind of the importance of heterogeneity, diversity in biology, and also that in two stories at least, that stem cells are heterogeneous and they drive tissue function. And then if anybody has ideas for collaboration with myself or anyone else, hopefully that you've thought about that during the talk. And just a big thank you to everyone who's helped, especially Lauren, who's been my partner in science for many years now. And since I'm making fun of her, I'm going to make fun of myself, and you can see me without a beard. But it's according, Dr. Feely thinks we only talk about science, but we actually do some things outside too. And again, big thank you to everyone in the Pomerantz lab and the Feely lab and all our collaborators and people who have been generous to donate tissue samples. And thank you to our grants and then also all the UCSF programs that are involved here. Thanks.
All right, thanks, Dr. Garcia. That was amazing and really impressive work. And let's see, Dr. Wostrak's hand up.
That was just amazing. I am so impressed with your work and just with your talk. I just want to say thank you for highlighting diversity. And I really like how you presented every slide with photos of your collaborators. I just want to say kudos to a great talk. As you know, I see a lot of patients that have radiation, and radiation fibrosis is a huge problem. We really don't have any solutions for it. And interestingly, it seems to set in about 7 months on average after high-dose radiation. And that's also a time that we're seeing similar problems in the nerves, and you might see radiation neuritis. Do you have any insight into the mechanism for the radiation fibrosis and why there's such a delay in seeing the clinical manifestations?
Yeah, that's a good point. I think, two things come to mind. One is that Andrew Brack would be a good person to talk to because he studies this directly. And then two, that the stem cells are mostly dormant, right? They're sleeping. And if you're not asking the muscle or the tissue to do anything, they generally are pretty okay, even with a lot of radiation damage. But then when you ask them to do something, they become active, they realize their DNA is messed up, and they either die through a process of apoptosis or they go down a pathway that's pathogenic. So I think maybe that, after 7 months, you just sort of reach a critical point where most of the stem cells have been activated by some means, and now you're left with the deranged ones, so to speak, that are causing a lot of pathogenesis. But it'd be interesting to study that over a time course or see what's happening to the heterogeneity of the stem cells. And I think also what plays into a role of this, there are stem cells that are radiation resistant, and maybe those are more responsible for fibrosis than some of the other tissue phenotypes, probably related to the nerve too. Yeah.
Dr. Garcia, thank you so much, Steve, for being an arthroplasty. I have a question for you, and excuse my ignorance. I want to go back to one of the early slides. We showed it wasn't a senescent cell, it was one step up from that, but a sleeping stem cell that went through a process of being triggered, awoken, if you will, and healing. But then there was another path that went to fibrogenesis, and then you went in, if I understand correctly, you sort of would try to elucidate what happens after it goes into that pathway. But do you have a clear idea why, at what point or what sort of injury triggers a differentiation in that? I'm not sure that was clear to me. How does it decide how to go bifurcate one way or the other?
Yeah, no, that is a question to still be answered in essence. But the real, I think the crux of it is that we don't know, but it seems that different injuries produce different phenotypes. So if in the mouse, if you do a certain injury, you can produce a lot of fibrosis, which is with cardiotoxin, which is like a typical muscle damage way that you can get a lot of tissue damage, but it only produces fibrosis. And then if you use glycerol, you can get only fat. So we know that the environment is very important to what is going on, and it's probably related to how FAPs talk to macrophages, and is one of the things that we're looking at in the lab. But we don't know why or what determines which way they go down, which lineage. But that's some of the things we're looking at too.
I'll have a follow-up question then.
Yeah.
I've heard of some work being done with epigenomics to try to trigger the awakening of these the one step up from that. So how does, and apparently it's quite effective in actually leading just down the healing pathway, not the fibrogenic or adipogenic pathway. Mm-hmm. Do you know much about that work? And
yeah, I mean, we know a little bit, or I know a little bit about it, but it seems like there are a lot of epigenetic changes that lead to senescence, and that's one of the big sort of funding opportunities through like the National Institute of Aging. A lot of people are looking at that through like P16 pathway, or like related to controlling the cell cycle with either methylation or adenylation. Another thing that has been shown is that these cells are more plastic than we think, especially at FAPs. They share common pathways with muscle cells. And if you give a certain molecule that releases an epigenetic change, you can actually induce MyoD, which is a master regulator or transcription factor of muscle regeneration. So you can sort of turn these cells into myogenic cells. Hasn't really been shown in humans, but that is another pathway that people are looking at, where you going to turn a cell into a more active cell that's doing something regenerative. But I haven't studied it personally.
So Dr. Collins had a question and comment on Zoom. So she said, great talk. Can you talk a little more about your piezo2 positive mechanosensitive population? Piezo2 is a great marker for sensory nerves. Would love your thoughts on what we could learn from comparing that population to others in your samples. Is there more adipose lipid deposition in populations with higher piezo2 positive?
Yeah, it does look like that. So the piezo2 population is interesting and something that I haven't had the opportunity to follow up further, but people in the lab have been very interested in that as well. The population looks like it is increased in injury, especially in the full thickness tear injury. And it seems that the protein is localized more to the myotendinous junction. And we know that FAPs do this special task, which is to help bind tendon to muscle. So if you think about it, you have to go from a very, you're taught this in ortho basic science, right? You have to go from a stiff tissue or ligament, like ligament or tendon, to a muscle, just a tendon to a muscle. And that seems to be a mechanosensitive area, and these cells seem to sit there. And it does seem to correlate with rotator cuff tears, where you do have a mechanical change in the biology of the organ. And that also is related to adipogenesis and fibrogenesis, but these cells don't seem like they have more adipogenesis. They seem like they're more of the myofibroblast type of cell.
All right. Any other questions or thoughts?
I have a question if there's not any in the room.
Yes, go ahead.
Beautiful talk, Dr. Garcia. I just want to congratulate you on leading the way or showing us the way here about using human tissues to do discovery biology. It's really impressive what you've learned, and it's of utmost clinical relevance since you're using tissues from people who are undergoing the care that's provided by this department. I'm excited to see more of that in our field. The questions that I have are, do you see any differences across the age spectrum of the patients that you're looking at? And then the other is, what can you learn about the niche cells that are providing support for this progenitor stem cell population from your single cell-seq? Have you learned anything already about the niche?
Yeah, good questions. The first one is, yes, there are differences, and Kevin Wang, who's one of the sports fellows, is interested in looking at that as well. So we're currently collecting more samples to look at to really highlight that, but it does look like there is a difference. The older patients seem to have more pre-fibrogenic and pre-adipogenic cells just at baseline. So they're more, you could think of it as primed to produce this degenerative phenotype. And work in the lab has shown that if you isolate cells from healthy adults versus elderly patients, that the cells quickly produce fatty fibrosis in the dish compared to their healthy counterparts. And then I think that the second one was the niche. I think we're also very interested in that, and we're looking at using some of these spatial techniques to look at that. One is with the MIBI collaboration with Stan. The other one is some of the work that Justin is doing with bioinformatics to look at the cell-cell interactions between FAPs and macrophages, as well as Schwann cells and the other resident muscle stem cell types like endothelial cells. So we're actively working at that too. And it seems like the FAPs are intimately involved with every single cell type in muscle, which kind of makes sense. They're like this intermediate cell that's acting to form a cohesive group to either have to maintain that functional niche in healthy tissue, and also leads to some of the pathology that you've seen when that heterogeneity balance is disturbed. So hopefully coming soon.
Hey, Stephen, great job over here. I think congratulations on what you're able to do in the lab, and also continue with your clinical years, and also the collaboration you have with all the people that you work with. So from a more clinical standpoint, as a shoulder surgeon, learning these repair processes is great. How close are we to bring this to clinical use, and what is the biggest impact you're going to see right now? Because, let's say for us, we clearly have a population of patients we weren't able to treat very well when they actually have fat inside the muscle. And we have to do replacement, which is a great operation, but still the complication with those. So what do you think the biggest application is, and how close are we?
Yeah.
What research you're doing right now?
Yeah, good questions. And the things that really matter, right? I think that we're probably not close, but I feel like there's two places that you can intervene. One is when you have an acute tear and this is going to develop a pathology, but say you can't get to surgery, so you have some drug to prevent the cells from forming fat or fibrosis. The other opportunity is that someone comes to you, they've already had a massive degenerative tear, they have a lot of fatty infiltration. How do you help that, and how do you reverse those chronic changes? And that's something we're working on in the lab right now too. Can we change, or can we influence that fat to either go away or to change into a more healthy phenotype with some of the work that Michael and Hannah and Christian are working on with changing white fat to brown fat? So I think, we're sort of beginning to understand why these cells make the decisions that they make. But as far as a real treatment option, all of the options we have are not good to give to humans. Like you wouldn't give a human a protein treatment. It would, we need to look at more of kind of these small molecule inhibitor type of based of therapies, probably, that are doing the same thing but not through a protein.
do you think it's more on injury prevention is what this research is going to lead to, or actually helping healing once the injury occurs? Yeah.
Yeah, I think it's more prevention. And I think we do have some things that we can do when patients are you have an injury and you don't want them to develop the chronic changes. And that could be preventing FAPs from dividing or proliferating. And some work is looking at like, how do we prevent that? One thing that we could borrow, maybe we need more studies, but if you look at heterotopic ossification, we think that's from FAPs as well, or at least I do. I don't know if it's been proven yet. But the way we've been traditionally treating that is with radiation, right, or NSAIDs. And we know that FAPs respond to both of those things, so maybe we should be looking at those in muscle as well.
You're very humble. You do have a CERM grant also that studies probably not practical ways necessarily to stimulate muscle, but we're not quite as far away as I think Stephen gives it credit for.
I'm the pessimist of the lab, so you guys are.
Speaking of which, how do you talked a little bit, especially at the start, of diversity and mentorship. How do you decide as you are transitioning from kind of that postdoctoral role where you're doing all the work versus you get all those people in that corner to do the work for you, and how do you transition to become this essentially your own independent PI, even though Shubhai and I pretend to be in charge of you?
Yeah, I mean, I think that it's tough to let go of wanting to pipette in the lab. I think that, but I think that hopefully we're both, the people that I'm working with are learning as much, or I'm learning as much from them as they are from me. And I think that as I've gotten farther along in the career, the transition is to thinking about the entirety of the experiment and how that applies to your global plan for your lab, and how we're effectively getting at what Dr. Mott was thinking about, like what is the end goal of this research and how we treat patients, and making sure that every experiment is falling into that and has a goal with a clear hypothesis and a way to move forward after each result.
All right. Well, thank you again, Dr. Garcia. Wonderful.